friends, welcome back. Today we will discuss with the third part of vaccination. Myself, Phoebe Paiva, Assistant Professor, Department of Zoology, St. Joseph's College for Women, Alapura. DNA vaccines. We know that vaccination is a simple introduction of a antigen or pathogenic organism into a host to induce the immunity in that particular host. But then DNA vaccination is a novel strategy of creating a vaccine without introducing the antigen. So how come immunity is developed? The It is developed because here we are introducing not the antigen but the DNA responsible for that antigen into the host. So this DNA for that for particular specific antigen is introduced into the host cell nucleus, whereupon it becomes the part of the cell's DNA, and as it is being expressed, the antigenic protein will be released. Now the host body possesses the antigenic protein and it is released gradually, and now the body will respond to that antigenic protein, and as a response, immune response elicit, they will release the antibodies, and also these antigenic proteins will also be responsible to activate the T cells or the cell mediated immunity. So here we can say that thus DNA vaccines are DNA sequence. Actually we are giving the DNA sequence of the antigen, and it is introduced into the individual to create the antigenic protein, and that antigenic, that in fact we could say antigen, that antigenic protein will activate the immune system in a normal way as a person do have an infection. So normally how a person in response to antigen, when an antigen enters the body, how it elicit or develop a immune response will be executed here too. And so we can say that this is a third generation vaccine. But the difference is that uh, when compared to a normal infection, here when we introduce the DNA, since it is not the antigen as such, when in the condition of introduction, they will not cause the development of symptoms, but they will elicit the introduction of the immunity. So here we can say it is a genetic immunization because DNA is given, okay? And DNA administration can be done either by injection, through liposomes, or by particle bombardment. So what is the principle? So the aim of DNA vaccination is to prevent disease, and by causing the expression of an antigen that stimulate the immune system. So here to reduce disease by introducing a DNA which is responsible to gradually develop into the antigenic protein, and by likewise it will develop the immune response. So when that plasmid DNA of the encoding protein is injected directly into the muscle, like for example if we are introducing into a muscle cell, that muscle will take the DNA and gradually it will be expressed into the protein. So DNA appears to integrate into a chromosomal DNA. So that foreign DNA or the antigenic DNA introduced can have two futures. That is, one, either it can become a part of the host chromosomal DNA, or second, it may remain outside the chromosomal DNA of the host as within the nucleus as a for a long time as episome form. We call it as episome. So at present, this DNA vaccination trials are going on, and it have been tried for AIDS, influenza, and herpes virus. So here we could see the pictorial representation. Here the viral DNA, we could see that viral DNA, it has been inserted into a vector, a plasmid, and this is introduced into the nucleus, and now it is in a episome form. That is, it is not integrated to the host DNA. So this is the host DNA, that is your DNA. So now it remains, in some cases it remain as episome for a long time, but in some cases this green part will be getting a part of the DNA. Whatever the reason, that DNA when expressed will form the mRNA. After translation will form they come out of the nucleus, they form the viral nucleoprotein. And this will be broken down into small pieces, and you could see that that will be forming that will be forming as a vesicle that will be broken down in the phagolysosomes, and a small part will be expressed by MHC and MHC class 2, and this will be uh, identified by T helper cells or the cytotoxic T cell. At the same time, some of them will be released out as naive, that is the antigenic protein which has not been broken down into pieces. Okay, so in a full condition itself, in a full form, they will be released, which will activate the B cells. So here, in one way or the other, both B cells and the helper T cells will be activated. That means both humoral immune system and the cell-mediated immune system will be activated. If it has been the T helper cell which has been activated because of MHC class 2, because of the small part on the MHC class 2, that will release some kind of signals which will activate cytotoxic T cells and the B cells and other phagocytic cells. So here you could see the diagrammatic representation, that is the gene of interest viral DNA, that is antigenic DNA. Actually, it's not viral, antigenic. Anything can be possible. It's inserted into the plasmid vector, and that is introduced into the nucleus, and you could see that those will be forming the mRNA, which will form the protein. Now, this in some cases, that complete protein will be released out, forming the free antigen, and that will activate the humoral response. Whereas in some cases, they will be broken down into pieces, and that will be expressed by a protein MHC class 1 or class 2, which will be activating the cell-mediated immunity. So advantages remains with the DNA vaccine is its simplicity, wide applicability. Large quantities of vaccines can be produced because we are not using the antigen as such. We just need a DNA that can be proliferated into any number. And here we can say that another advantage is that the protein will be expressed in the host in the normal way. As I said earlier, as a person get an infection, as a person get the exposure, similar way this protein, once it is formed, they will be expressed in the host in the natural form. And here we can say next advantage is that both humoral and cell mediated immunity has been activated. The third one is the next one is that they have a long immunological memory. And very important practical application is that refrigeration is not needed, so that the amount of cost required for handling and storage of the other DNA, other vaccines can be reduced by this. So greatly lowers the cost and complexity of delivery. It can be taken to the interiors of the rural areas. Thank you.